Hello and welcome everyone for another webinar from our Open Neuroscience seminar series, which is still part of the Worldwide Neuro Initiative. So this series, as you know, it's about open source and open science tools for neurosciences. And today we are very pleased to receive Andrea Giovannucci from the University of North Carolina. Andrea obtained his PhD from the Autonomous University of Barcelona and the Artificial Intelligence Institute in Bellaterra in Spain. He then joined the Factorization Institute as a machine learning data scientist, and then he became a postdoctoral fellow at the Princeton Neuroscience Institute. He is now a professor in neural engineering within the Department of Bioengineering at the University of North Carolina, and there he manages his own team, the Neural Engineering Laboratory. Today, Andrea has kindly accepted our invitation to talk about CaImAn. It's an acronym for Calcium Imaging Data Analysis. It's a framework for the analysis of large-scale one-photon and multi-photon calcium imaging dataset. This particular software provides efficient routines for solving pre-processing problems such as motion correction, source separation, spice deconvolution, etc. But I will not say anymore. I won't spoil the fun, so I will let Andrea demonstrate himself the beauty of his system. So thank you, Andrea, for being here with us today.
Thank you so much for the kind introduction. And should I share my screen? At this point?
You can go ahead. Have a good.
I would like to thank your team, not only for inviting me today and sharing this package with you, but also for the fantastic effort you're having. I really loved your webpage. I love the way it's organized. I love that there is a small movie that accompany the explanation of each software. I think this is going to be key for many people to basically access this software and lowering the barrier. Okay, so
that's a goal.
Sorry?
I was saying thank you. So we hope that's a goal.
Okay, so let me go straight to the point. So today I'm going to talk about CaImAn. And just to give you a little bit of an overview about this package, the story of this package is now like 8 years old, approximately. It was started at the Simons Foundation in the Flatiron Institute in the Center for Computational Biology. And since then, it's kept, we keep it maintaining. My laboratory is adding new algorithms, and the Simons Foundation, Flatiron Institute, is maintaining the software code and making sure basically the machine works and is well tested. What is CaImAn? CaImAn is a general purpose package for the analysis of fluorescence imaging at a cellular level. And at this moment, this basically manifests in two types of analysis: voltage and calcium imaging. There are different modalities in which these indicators can be imaged. You can do one-photon imaging, which allows you to go deeper into the brain and have freely moving animals. You can have multi-photon imaging, you can have volumetric, both one and two photon imaging in this case. And you also have different type of modality in which you can analyze data, online and batch, as I will detail later. What's important about CaImAn is not just about the software, because one thing is to develop algorithms, and the other thing is to make it really work for the big community. So these are really two different things. And I would say probably the effort of making available to the big community is much larger than the initial effort of developing an algorithm. For that, all the things that you can see on the right, we need infrastructure that, runs from, testing to packaging and installation. Then you want to support users with different communication channels based on GitHub or others like this effort that we are carrying out today, in which we provide tutorials, but we also organize workshops, and we try to keep and update documentation. And finally, slowly we're going also towards establishing CaImAn as a one-click software, meaning you are going to start, for instance, a notebook in Google Colab and start your analysis there, because a lot of problems, as you know, comes from the installation. All these things, of course, require effort, and it's not an effort that can be carried out only by one person. Not only a set of team members are required, but also the community is fundamental. And we have about 43 collaborators, contributors in the CaImAn repository now. So I would like also to thank the open source community for the big help they are providing there. Today, I'm going to focus on two specific portions, if you want, of the capabilities of CaImAn. One is the analysis of calcium imaging in the batch modality, And the second one is the analysis of voltage indicators, which is also batch, and it's one photon, because most of the indicators available today are still compatible only with one photon and not with two photon imaging. Let me tell you a little about the two modalities that we're discussing, calcium and voltage imaging. Many of you are familiar with this idea, but very quickly, what's the power of multi-photon imaging is that you can focus, you can optically section the brain, go at some depth into the tissue, and then see what's happening there. And when I see what's happening there, you can really literally see the activity of the cell turning on, as you can see in this example. For instance, in this case, you can see the soma of neurons in the hippocampus. These little rings that becomes brighter are somas. Here we have dendrites. These diagonal bands that turn on are dendrites in the mouse cerebellum. And then you might have actually a mix of the two. In this case, you can see both neurons turning on, but also processes that turn on. So what you would normally do to extract the activity of calcium, you take one neuron, for instance, like this one, then you take the average of these pixels, and you will observe a fluorescent signal that changes with these dynamics. This dynamics reflects the intracellular calcium level of these neurons. So there is an indirect, if you want, measure of the activity of neurons. And what you want to do now from these fluorescence traces, go back and resort to spikes. So at the end of the day, the results of your analysis of this type of movies is going from this raw movie to the spikes, the activity of each neurons. The second type of imaging modality that we're dealing with today is one photon imaging, but only for voltage in this case. But just to give you a little bit of the flavor, when you compare the calcium imaging movie here on the top with the two photon imaging movies that you saw before, you see that it's much blurrier. There is a lot of other things happening in the movie that don't seem to be related directly to the activity of single neurons. And this is because you are integrating light coming from a large chunk of tissue. So the background, everything that is non-neuron here, it's much more complex than in the other case. The other case is, apologies. The other case is voltage imaging. Voltage imaging, this movie is actually running. You don't see the activity, but actually this is a movie that is running. And the signal to noise ratio is much lower. So we require a little bit of more advanced methods to be able to extract activity from these movies. A big difference that you want to make when you analyze data is between batch analysis, that it means you have all the movies available, the whole movie available at a single point. For instance, after you've done your experiment, you can run the analysis overnight and then collect the analysis the day after. This is called batch analysis. Another type of analysis is online analysis. In online analysis means that you don't have access, either because you cannot load it into memory or because it's an ongoing experiment. You don't have access to the activity at each frame. So what you want to do, you basically want to retrieve you want to be able to approximate the activity of the brain at each time point, just looking at a single frame. And we call this online analysis. Today I'm going to focus only on the batch portion of this analysis. And either we can do another open neuroscience workshop, or you can refer to the to the papers and documentation if you want to know more about the online analysis. No matter what you do, online, offline, batch, online, batch, voltage, or calcium imaging, there is a set of universal preprocessing steps that needs to be carried out to extract activity of neurons. And this one starts with correct information, then localizing where neurons are, approximately. Then you want to clean up this data. You want to increase the signal-to-noise resolution. And in this case, both in calcium and voltage imaging, something that it was mentioned before in the introduction when we were discussing, is that there is contamination from other sources. So, for instance, if you look at this movie on the right, and you naively segment each of these neurons and take the average of the pixels, you will see that there is contamination between the two signals. Basically, when the red neurons fires, you would see a little bit of that activity in the yellow. So this problem is not trivially a segmentation problem, but it's what we call a source separation problem. So once you cleaned up your data, that looks much nicer, you cleaned up your components, then you want to extract spikes. And you can extract spikes in calcium imaging with a process that's called deconvolution, or extract spikes in voltage. In the case of voltage imaging, there is a further operation you can perform, which is extract the subthreshold oscillations. That's one of the things that would make actually voltage better than calcium imaging, is that not only you have access to suprathreshold activity, but you can have access also to subthreshold activity of neurons. Cool. Let's start, with the idea of motion correction. What does it mean, motion correction? If you look at the image, the movie above, you will see that in the left you have this movie that moves around. What is the problem of having this brain moving under the microscope, right? The problem is that most of the algorithms that are used for processing these images assume that each pixel will always correspond to the same spatial location. So what you want to do is now correct this movie and make sure that each pixel will always represent the similar spatial location. So you have a mouse, for instance, is running on the treadmill, it moves around, the brain is mushy, and this will mess up the results of your algorithm. There are two types of motion correction. One is rigid, that means you simply shift your frame to align it to a reference image, or a non-rigid approximation. If you look at the movie again on the left, you will see it's not enough to simply shift a template to match what's happening in the movie, but there is a non-rigid. There is a movement that is not rigid of your frame, and you need to compensate that. What is CaImAn doing for this? CaImAn is partitioning the field of view in small squares, patches, and then is motion correcting each of them. And at the end of the day, it's basically interpolating the shifts that have been estimated for each of these little patches. To be a little bit more concrete about the parameters that control this process, you have a max shift parameter. I'm highlighting here just the most important parameters here. But if you want to know more, you can look at the documentation. But the most important is the max shift. That means what is the maximum shift of your field of view that they are expecting. Okay, so for instance, if from the beginning of the experiments or to the end of the experiments there is a slow drift, you want to have a larger number for this value, and then the patch size. So you can consider this like basically the size corresponding to the size roughly of each of these patches. And what happens if you have many areas which are dark? When you try to motion correct the dark area, you will not have a good result. So there you have, in some sense, a trade-off between robust motion correction as you make this patch bigger, and the fact that you will not be able to capture as much the non-rigid aspect of the motion. Once we are done, we create a memory map file. What is a memory map file? A memory map file is a file that can be accessed directly from the hard drive without the need to be loaded in memory. So let's say that I have a 200,000 frame movie and I only have a 16 gigabyte desktop. I can still be able to access different frames by using this type of infrastructure. Now, this infrastructure needs to be, it's very powerful, but it needs to be used correctly. So in this case, we save this file in what's called F-order. F-order means that reading frame by frame is efficient. If you try to read a single pixel, for instance, for all the 200,000 frames, this will be very, very slow. So be mindful, if it's an F-order, then you are allowed, if you want, to read frame by frame, but it's very discouraged to read pixel by pixel. Motion correction is parallelized, and because the whole idea is that you want to use a small machine to analyze a data that is much larger than your available RAM memory. And to do so, you basically chunk your movie in time. So you get, for instance, 100 frame chunks of 100 frames. Each of these 100 frames is motion corrected by a different process. And then the result is stored into a memory map file. So this memory mappable file can not only be read, right, without loading in memory, but can also be written on without loading in memory. So this basically allows you to have a very, very large file that can be corrected by a small machine. Okay, as I mentioned to you before, this format, data format, the memory map format, is easily readable in frame by frame. But what we need for the subsequent analysis is actually something that is readable pixel by pixel. So loading a chunk of pixel instead of a chunk of frames. So we transform this into what is a C-ordered file. And everything is done in CaImAn automatically and in parallel. But I want you to be aware of the creation because of these big data sets that are gonna, fill space in your hard drive, can be, of course, erased afterwards. But also the fact that you want to know how to use them properly in case you want to customize software. Perfect. So we are now left with the original file and 2 copies on the hard drive of this file, one that is easily readable frame by frame and one that is easily readable pixel by pixel. Apologies. Okay, so now let's switch to the second portion of the analysis that we discussed, which is separating the sources, segmenting neurons, and making sure that the activity is demixed. To do this, we use a framework called Constrained Non-Negative Matrix Factorization. And the basic idea is rather simple. You take a movie and you try to express it as the sum of different components. And these components will have 2 types of natures, if you want. Components that are related to neurons, like we call them A and C, and then components that are related to background, so to contamination that comes from the neuropil, for instance. A and C are 2 matrices. The matrix A represents the spatial components, that is, in this case, the spatial footprint of a neuron, and the C matrix represents the temporal components. Okay, the time at the activity of the neurons, the fluorescence. The same happens, of course, for the background components, which the B represents space and F represent the time varying aspect of the background. Formally, this is expressed as a non-negative matrix factorization problem, as I was telling you. And in exactly what I explained to you before, you try to use AC and BF to approximate Y. So you are minimize the difference between the 2. There are some constraints on these components. For instance, A will be sparse because we don't expect to have many pixels occupied by neurons, just a few of them. Most of them will be black with just some white pixels. And then we have a non-negative constraints. So the non-negativity constraints is very important. It's basically what helps you to demix signals. It what imposes the fact that you can only sum the activity of these components, and you cannot subtract it among them. So this is exactly the trick that allows you to separate the neurons. And in more detail, an example here in which you have a movie, you vectorize the movie from height times width times number of frames by number of pixels times number of frames. Then you express them as the sum of the multiplication of different smaller matrices. And when you look inside these matrices, I anticipated before, the column of A, when it's reshaped back to the original shape of the movie, of the fields of view, will look exactly like the shape of a neuron. And the C matrix, if you take one row, it will look like the activity. And why is this, solving the problem that we anticipated before? When you solve this, the negative matrix factorization, why do you really get back the activity of neurons? Is because now if you try to make the outer product, basically if you multiply these neurons, and then you reshape to the original movie, you actually obtain the activity only of that neuron. Okay? So these methods inherently really helps you to separate these activities by merging together, by combining pixels that are highly correlated. And the same can be said for the background components. And if you take B, you will see that and the spatial component will look like exactly like the average, if you want, of the background, and the time components will tell you how the background evolves. And if you look at it, you can clearly see that there is a lot of activity of the background. See how this changes? And this, if you don't capture it, it will all go into the signal of the neurons and might confound your final results. One word about the background. The background, doesn't have any constraint. You say it's not like sparse like the A. It can do whatever. It can go everywhere and capture whatever. So you want to keep the numbers of these components, can be 1, 2, or 3, very low. Because if you start increasing it, it will start sucking the activity of neurons. And you don't want that to happen.
Excuse me, Andrea, do you mind if I pause you for a minute? Do you mind if we ask you a question about the neuropil?
Please.
Okay. Sylvia, go ahead.
Yeah, hi. I just wanted to understand a bit better what assumptions you make about the neuropil. Maybe you will just talk about this now. But, how many components, are they kind of spatially constrained somehow, or yeah?
The assumption of the neuropils are very basic in this case. This model basically tells you if you find a set of pixels, okay, which are highly correlated in time, okay, then you basically express the activity of these pixels that are highly correlated as this product. So the assumption is that there is a set of pixels that go up and down together. And, you can have 2 different places in which this happens. You can have 3 places in which it happens. So this assumption, for instance, just to give you a panoramic idea of that, this assumption slowly breaks down, right, as you have more complex background. And, for instance, if you look at one-photon imaging, this assumption of having this type of low rank, if you want, representation, low rank means few rows and columns expressed in the background. With one-photon imaging, this breaks down because it becomes so complex that this doesn't hold anymore, and you need more powerful representations. So,
but I already don't understand how you distinguish between the neurons and the background. So neurons are also correlated, right? Maybe I missed something.
Neurons often, they turn on by themselves. So basically, if you look at the correlation between pixels, example, right? And you will see that there is a set of neurons with a set of pixels within the neurons that are highly, highly correlated. And they're a bit less correlated with the background, right? Because the background will not go up, down exactly the same way. So that is the assumption. Of course, if the background goes up and down exactly in the same way that the group of neurons, you get troubles, and actually this system fails. Example, if you photostimulate a large set of neurons, okay, all at the same time, they will end up in the background. Make sense what I'm saying?
Yes, okay. But so, yes, I think we're getting there. But when do you put a column into matrix A, and when does it go into
okay, this
matrix
goes a
B?
little more into the details of the initialization. I can go a little bit more into the detail if you want. So what do you
I'd
do?
like to, if that's okay.
It depends on time, but we are there for this reason. So what happens during the initialization? During the initialization, you basically you try to pick up neurons one by one. It's really a peeling off process. You take one neuron, you estimate its activity, doing a rank 1 approximation of a little square, okay? And then you remove the activity of the neurons from the movie, and then you go with the second, then you go with the third, until a number that we see that later you specify. At that point, whatever is left in pixels is used to estimate the backgrounds, 1 or 2 components. And that estimate is also done by doing see it as a PCA with 2 components, but non-negativity. So you say, okay, how can I capture the major direction of variance of this background? 2 components, only the pixel that didn't belong to neurons. Okay? And this
all right. One question. So how do you make sure when you take the neurons, it's only the neurons rather than the background in the first peeling off process? There must be some constraint, I guess.
Well, the way you pick it up is by doing this local rank 1 approximation. So first we make sure that there is something by looking at the variance of these pixels, and then we do a rank 1 approximation. If the rank 1 approximation fails, basically doesn't give you something that looks like a neurons, it will be removed later.
I see, okay.
it's very important what you say, because there is a refinement process that needs to take care of this, right? This is just an initialization. But then you want to be sure that during this refinement, both you make sure the neurons, are well expressed by the spatial components. And secondly, you want to be sure that whatever is contribution from the background is removed from that. So what do? You go back and forth between, the spatial components, temporal components, background, and neurons, back and forth, and until you converge. Okay. Of course, this doesn't guarantee you that you will get the right results, because the assumption, as I mentioned before, is that not all the neurons are super correlated with the background. If the whole neuron are super correlated with the background, this breaks.
Okay, thank you.
My pleasure. This is a very good question, and a complex one, actually, to tackle if you want to solve the problem of highly correlated neural activity. Cool. So this is what a movie that has been analyzed look like. Okay. Imagine here on the left, you have you found all these neurons, the spatial components. Here you find the temporal components. And now you can do some nice visualization to see how your analysis performs. On the left, on the top, you have the raw movie. Here you can use the components and the background that you found, as I showed you in the previous slide, to reconstruct the movie. And what you get is basically the noise version of the original movie. You can see it here. You can also visualize just the neural activity. So basically, you remove all the contribution for what is the estimated background and show the neural activity. But the most interesting representation is this one, which is the residual. The residual means you remove the activity of the estimated neurons from the original one. So if you did a good job, in this case, what you will have is something that looks like noise. If you get something that doesn't show much structure, then it means that you did a proper analysis. Okay, let's talk a little bit about the computational infrastructure underlying CaImAn. The computational infrastructure is, as I explained to you before, there is this motion correction and memory mapping that creates the files for us. And then we need to find a way to apply this non-negative matrix factorization algorithm. And I was mentioning to Silvia before, this is a very iterative process. But the way we do it, we started, in small patches. In small patches, does it mean if you try to solve the problem of finding neurons on the big field of view, this is very similar to the problem of finding neurons on a small patch if you take care of what happens at the intersection of these patches. So if you do this, you can actually do the assumption that I can separately deal with each of these patches. And actually, in some sense, it's also more interesting, coming back to the background, if you have complex background that is local, this type of analysis will be able to capture a more local background. So what do? We partition our original movie in many small tensors, okay, like these ones. We analyze each of these tensors using a different process. Now, I remind you that we had the memory map file. What does it mean? Again, they don't need to load in memory the whole thing. They just read what's needed for them to work. Each of them will need the appropriate portion of the movie, process them, and at the end, we will take care of merging neurons that were in between the two. For this, we basically look at the amount of overlap and the correlation between the signals of the two detected neurons. Importantly, if you run this on a high performance cluster, what you can do, let's say that you have 200 nodes, you can separately analyze each of these chunks on a separate node, which make the analysis very, very fast if you have the right computational infrastructure. Let me go through some of the parameters. And again, here I'm just going through the parameters that I think are fundamental to get started, but it can be refined by looking at documentation. So one is RF. RF is half of the size of this patch. So let's say if you have 15 RF, it means that you're looking at patches of 30 by 30. The second parameter is K. K is the number of neurons that you expect to find in that patch. And Sylvia, again, this answering your question before, we are a little bit generous here. So we put a little bit more than the number that we would expect in general to really clean up every type of possible signal we could find in these little patches that is not background. And this is the number that tells you how many neurons you want to try, you expect to find. And finally, G-sig is half of the size of the neurons that you expect to find. So let's say that your neurons are 10 pixels in diameter, then you want to put 5 here, half of the size. Other interesting parameters are deconvolution, P. I'm not gonna go into the details of deconvolution today. It's a way to extract spikes because I think we don't have time for that. But just to give you a rule of thumb, you want to use P equal 1 when you have a fast rise time. So for instance, you're using a fast indicator and it takes just 1 frame to go to the peak, and P equal 2 if you're using a slow one that takes some time to reach to the peak of the calcium transients. G and B is the number of background. In general, you want to keep it 1 or 2 for the reason that I explained before. And merge threshold is the correlation that the minimal correlation that we are accepting between components to merge. Okay, so if you have components that are merged incorrectly, then you want to raise this number a little bit. 0.8, 0.9, even 0.95 in some cases. Okay, so as I told you, what we are very, very generous when you initialize this algorithm. So we actually asking the algorithm to find more components that one could actually, than they actually are in the movie. So we refine the solutions with some tricks, and our tricks are the following. So one is looking at the signal to noise ratio. So you have the parameters, for instance, mean SNR, which will add components only if they are above a given measure of signal to noise ratio. In general, this is 2 or 3. The second one, it's called the spatial correlation, which is the correlation between the spatial component and the average of the frames in which the neuron was active. The assumption here is that if we did a good job and it's not noise, there will be a high correlation with what the components look like in the NMF and what it looks like in your movie. And finally, we use a convolutional neural network that decides what looks like a neuron from what doesn't look like a neuron. This is an example of a training set, and this is an example of the test set. What you see is that the fact that it's not selected by this, if you want, refinement step, doesn't mean that doesn't contain signals. These points are probably some axons or dendrites that were just cut off by the imaging plane, but still carry a lot of signal. So pay attention if you're interested in this type of signal. You don't want to use this refinement process with a set of high parameters. So 2 are the parameters here. We use 2 parameters for this one, which is the threshold to add a neuron. A neuron, if it's as a probability of looking like a neuron, larger than 0.99, then we add it. But we also have this one, which is a very strong constraint, which remove from the final set of neurons every neuron which has a probability of less than 0.1 to look like a neuron. So again, pay attention to this parameter. You might want to lower it if your results get worse. And this is an example of what the solution refinement does. You know, on the left, we are very generous. We select a lot of neurons, but then in our final set of accepted components, we only keep some of them that we think are more promising. Of course, the result is not perfect, but is much, much better than what we had on the left. Okay, so now I'm going to switch gears and go to voltage imaging. I don't know if there is any question about calcium imaging that we want to answer. And also maybe a quick question about timing. How much time do I have left?
You have a good 20 minutes.
Okay, that's fantastic. Okay, so I'm moving forward. So this is an example of voltage imaging movie. And this is the state-of-the-art Voltron, and you don't see much in the movie. If you start removing the background, the baseline, then you start seeing very interesting activity. Again, it's not comparable to the clarity of the signal that you get from calcium imaging. So we did is develop some tools, some new instruments that will enable enhancing the signal-to-noise ratio of this type of movies. Why do we want to use voltage imaging? Well, first, it gives you the memory potential, so subthreshold activity. Second, the sub-millisecond time resolution that it provides to the spike resolution, the dynamics of the signal is such that it's not like calcium. You know, when we see a peak, it's a real spike. That's the real thing. There is no doubt. And finally, it allows you to study subthreshold dynamics at the level of population. But there are some challenges. One is that it's only one photon, which adds to the fact that there is a low signal-to-noise resolution, signal-to-noise ratio, and the fact that by themselves, actually, they are a low signal-to-noise ratio type of probe. But recently, they enabled imaging a large number of neurons, 50, 55, and more, okay? And for each of them provides these, you know, traces that I would say that are very, very promising in terms of signal-noise to ratio, even at the level of multiple neurons. And what you will end up doing for these traces when they look good is to extract the spikes, these peaks, and extract the subthreshold movements, the subthreshold dynamics of the voltage of these neurons. How do we do that? So I'm not going to go into motion correction and memory mapping because they're very, very, very similar, except in smaller variation to what I just presented. I will just very quickly focus on the segmentation. Since it is, in order to image this type of neurons, it's necessary to have sparse expression. That means not very many neurons in the same space, in the same, let's say, portion of the field of view. We decided to use a deep learning approach to initialize. So basically, you can look at the movies, see where neurons are, draw a little box around them. And for doing this, we use a Mascar CNN, which is a state-of-the-art networks for image segmentation. It turned out to work very, very, very well. And then we complement with a small visual interface the results. So you can add or remove from the results that you found with the network to refine. And, you know, if you really are computationally savvy and you have your own labeled data, you can actually retrain the algorithms for your own purposes. Let's talk a little bit about the spike trace denoising and spike extraction. The problem is different from what we saw in calcium imaging for a set of reasons. So one is, of course, the general signal-to-noise ratio, but also the fact that the signal is negative. So the polarity of many of these indicators is negative. That means when there is a spike, the signal doesn't become brighter, but it becomes actually dimmer. So this already poses some challenges. Secondly, the structure of the background is a little bit more complex. So to do this, we basically undergo a set of pre-processing steps. One is we high-pass filter. One big problem with voltage imaging is also the fact that the photo bleaches. So you might start with 100% of the signal, and you end up maybe with 30 or 40% of the original signal. So we do high-pass filtering to normalize in some sense for this, to the trended signal. Then we try to remove the background by using singular value decomposition. That means we simply try to regress from within the neurons all the activity that is similar to the background, and we remove it from it. Once we've done this, we select a few number of spikes, the most evident, okay, let's say 10, 20, 50, and from them we build a template. So something, a shape, a waveform that looks like a transient in this voltage fluorescence movies. Once we've done this, we can do what's called white and match filter, that is basically computing a normalized cross correlation of this template with a trace, but paying attention that the noise, basically the the effect of the convolved noise is smaller. Once we have this new trace that has been computed, we try to fit it to the pixels and find the pixels that best explain this trace. So we go back and forth again from the finding the spikes and then refining the spatial component so that it basically gives you the best, highest possible signal to noise ratio. And this is what happens, you know, when you look at the movies now that have been processed with VolPy, which is the name of the algorithm. So the original, removing the background, and now here is what you get with VolPy. So VolPy really clean up significantly the data, and now spikes becomes much, much clearer. What are the important parameters if you want for VolPy? It's actually pretty robust. Really, the most critical, if you want, parameter here is the initialization of the spikes. So, and if you have the right threshold for that, and you want to select a set of spikes, how do you know, if you have few spikes, how do you know that you're selecting the proper threshold? So sometimes it's required to have some manual tweaking of these thresholds. But this the adaptive, that is, used by this P norm, in general will give you most of the traces with the appropriate threshold. So you might want to post-process a little bit few of the traces, but in general, the results are pretty positive. Okay, so here I am at, at the summary of the talk. So today we discussed a little bit of calcium imaging in the modality of multi-photon. I discussed how to motion correct in the non-rigid and rigid case. I gave you a little bit of the panoramic, what's the computational model underlying the separation of signals from overlapping regions and background. And then I switched to to single photon voltage imaging, and I discussed what are possible methods that are now developed by us to analyze voltage imaging data. Looking forward, especially in relation to this audience and things we want to improve on, are user training. So we are planning on organizing in the spring, probably late spring, a workshop at the Simons Foundation for CaImAn, so stay tuned on that one. And my laboratory in particular is interested in providing fast feedback on experiments. Again, I don't have time to go through that type of algorithms today, but what I think is missing in the area is feedback on ongoing experiments. So if you remember electrophysiologists 35 or 40 years ago, they were basically listening to spikes. And, while they were doing an experiment or changing something, they had immediate feedback about what's going on. If you think about the modern experiments with imaging, you are lucky if you're able just to keep up with what's going on, in terms of software and hardware. But what would be needed actually is some way, visual or audiovisual, to basically communicate what's going on your experiment in real time. That's why we are now developing algorithms that very quickly can extract the activity of the brain and give you, an idea of what is the brain state. Okay, and in this sense, we are working in two big branches. One branch is just basically extracting and know what the activity is and visualizing it, but also some way to reduce the dimensionality, and highlight to the final user what is an important thing that is happening in your experiment. And finally, we are working with, for instance, light sheet data from brain organoids or zebrafish, which now are 600, 700, 800 gigabytes, and it takes a long time to analyze them. So we are also trying to develop tools that can, that they will manage to analyze this type of data sets within a reasonable amount of time. Our target is about 1 hour, 2 hours. That said, I would like to thank all the fantastic researchers at the Simons Foundation and Flatiron Institute, Eftichios Pneumatikakis in particular, was been the co-founder of CaImAn with me, but significant contribution has been recently, has been actually since the very beginning, from Johannes Friedrich, from Pat Gunn, and Mitya Chlosky, which was the group leader at the Simons Foundation when we started the project. If you want to find the repository, you both have a link that it was probably provided by the team, but also you can find it on GitHub and in the paper from 2019 in eLife that describes in details everything I discussed today about calcium imaging. And about VOLPA, I would like to thank two very, very talented trainees, Changjia, which is a graduate student, second-year graduate student here at UNC Chapel Hill, and Cynthia Dong, that was an undergraduate, and now she's working in AI. But they really did a fantastic job for VOLPA. I would like also again to thank you for inviting me, for your effort for the open source community, and you all guys for the attention. Thank you.
Thank you a lot, Andrea. That was very intensive. Thanks a lot for all this work. I can say that we're really looking forward to any kind of collaboration or workshop you're going to advertise, or going to organize. We're going to keep looking for that. We're going to keep advertising it. Before we move on, I'd just like to remind our audience on every platform that if they want to ask questions, they can use the multiple chats at their disposal. If they want to join us on the Zoom, they can do that now, and therefore they can start exchanging with us and asking questions. While we're waiting for the first questions, I'd like to remind you all that you can find, of course, Andrea repository on GitHub in the link below. You can also find it on our own repository on Open Neuroscience, and you will find as well other project there. So that being said, is there any questions? I have one from Andrea from the University of Sussex. It's kind of general. Are you and the CaImAn team in contact with the Miniscope community? It seems that it would be great to get the two of you collaborating on the project.
We definitely are in contact. We are in contact with Denise Kai. We are in contact with Daniel Aroni. So we were invited to the Miniscope workshop this year, in which we discussed a little bit online analysis. They also have a very nice effort ongoing there. It's called Minion, which it's trying basically to combine much of this effort, but with more emphasis on one-photon imaging. We definitely are in contact. We try to develop tools in parallel for the community that can cover basically different bases. But yes, definitely, it's probably gonna be the most important segment of users that needs to be covered in the next years, because it's exploding. It's not like a two-photon imaging setup that costs $200,000. With a few tens, maybe $10,000, you can establish an experimental platform, and then at that point, your problem is just analyzing data.
Thank you for that. We have a question from Sylvia. Do you want to ask it yourself, Sylvia?
Sure, I can do that. I'm using so far SweetToP and wanted to ask you what the difference is. I don't really know the details, but it looked relatively similar. And is there any reason we should switch to CalmAn? I mean, I haven't heard about this new thing that you were saying about during the experiments, looking at the activity. That sounds very interesting. But in the post-processing, what do you think?
In general, I'm a practical person, and I'm reluctant to suggest to people to change their experimental computational pipeline. That's first thing said. In my experience, also, it seems to be more important what the parameters, the refinement of the parameters of an algorithm than the algorithm itself, okay, in terms of final outcome. That said, I think CaImAn is slightly better at dealing with contaminated signals. So basically, when you have a strong background or when neurons are overlapping, you can get a little bit more signal out of CaImAn, because the underlying model, as I told you before, is such that will separate these signals. But on the other end, for instance, when the non-negativity assumptions fail, then SweetToP does a little bit better. Again, I'm reluctant to suggest to people to change because there's a big cost associated to change such a big things, especially if you have a pipeline that is established. Now, if one wants to switch to online analysis, then that doesn't exist in SweetToP to the best of my knowledge. So I think CaImAn is the only one that provides this online support, which you might look at what happens in the experiment in real time. Still, it's not really keeping up with the real-time aspect of the experiment because of, depending on your hardware and how you optimize the algorithm. But, just recently we came up with a GPU algorithm that actually goes pretty fast, up to 300, 400 Hertz. And that should solve also that problem. So my answer is, don't change it for the batch analysis, but if you want to do online analysis, you might have a look at CaImAn.
Cool. Do you actually have a GUI like SweetToP where you can manually?
So no, not like this. I think SweetToP has a very nice GUI. I think it's beautiful. Now, what happened is we just received funding from the Chan Zuckerberg initiative to integrate CaImAn with Napari. So I think that we are going to have a very, very nice interface, in the next 6 months. That's going to be, again, probably will be more oriented towards this idea of online experiments, but we'll cover also very nicely the batch case. So we do have, I think the one that is in SweetToP is better.
Thank you.
No problem.
Great. Is there any other specific question? I will have plenty, but this technical, so maybe not worth of interest. I mean, at least for the live stream, at least. No. All right. So thank you, everyone. Thanks, Andrea, for being with us today. Once again, I can only encourage people to visit your repository to read the paper and maybe try CalmAn themselves. It's actually nicely outlined. And yeah, I say just give it a try.
Thank you so much. Thank you so much again for your effort. I really appreciate that. And, I hope the community also will reward you.
I hope so. Just stick with me a minute.
Yeah.
Okay. Goodbye, everybody. And we are offline. Okay. So I just